Hello viewers, welcome to the class 11 biology class. Today's lecture is on the mitochondrion. Mitochondria, as we call it, it's the plural number mitochondria. Now, what is the mitochondrion? The mitochondrion is actually the subcellular or subcellular organelle, which is subcellular means it is present inside the cell. Okay, so it is being governed, its functions are being governed by the cell as a whole, and it is a subcellular organelle. Okay, as I had discussed previously about the endoplasmic reticulum, which is also a subcellular organelle, and today I'll be talking about the mitochondria. Now, the mitochondria are also known as powerhouses of the cell. Why are they known as powerhouses? Do they generate power? Yes, they do. How do they generate power? What they do is that these convert the potential energy of the foodstuffs. Okay, what are the foodstuffs that we consume? We consume proteins, we consume carbohydrates, we consume lipids, fats, etc. So these convert the energy, the potential energy which is stored in the foodstuff into the kinetic energy form, which can be used by the living body of which the cell is a part, which houses the mitochondrion. Okay, now what are these subcellular organelles mainly responsible for? They are responsible for aerobic respiration. Now, we know that respiration can be anaerobic, which does not use oxygen, and aerobic, which uses oxygen. So, I repeat, the mitochondrion is actually responsible for carrying out aerobic respiration. We will talk about this in a little bit detail, though this is not a chapter of respiration, this is just a chapter of this subcellular organelle. Okay. Now, where are they present mostly? First, let me tell you that they are absent in the prokaryotes and they are present in the eukaryotic cells. However, we all know about the red blood corpuscles, red blood cells. The red blood cells, when they mature, they lose the mitochondria. There is no mitochondria in the mature red blood cells. Otherwise, they are just distributed all over the cytoplasm, or they are concentrated in those regions which require the maximum use of energy, because they are the energy suppliers, and that is the reason they are known as the powerhouses of the cell. Now, the number of mitochondria, it also increases where the cellular activity suddenly becomes more. Suppose the germinating seeds. Definitely we can understand that the germinating seeds will be requiring more power, more energy, more energy in the form of ATP, which we'll be talking about it a little later. So this is what is being produced in the aerobic respiration, which takes place inside the mitochondria. So the places which require more energy has got more mitochondria. Okay. Now, when we are talking about the ultrastructure of the mitochondria, we can see in the diagram which follows, in the diagram which is coming together along with, we can clearly see that the mitochondrion has got a double membrane. Okay, a double membrane. This entire this organelle is enclosed by a double membrane. The outer membrane is actually it is connecting. It is actually the outer part of the outer membrane. Okay, the outer part of the outer membrane is facing the cytoplasm or the cytosol of the cell of which the mitochondrion is a part. Now, the inner membrane, we can see, is thrown up into a number of folds. It's clear, I think. It is thrown up into a number of folds, and these folds have on them, there are, I've marked it with a green color, some elementary particles, or they are also known as oxysomes. Okay, talking about the outer mitochondrial membrane, it is actually isolating the mitochondrial contents, whatever is there inside the mitochondrion, from the cytosol. It is smooth and it is also connected with the membranes of the endoplasmic reticulum. We had studied about the endoplasmic reticulum a few days back. This outer membrane is actually freely permeable to molecules which enter the mitochondrial matrix, and it also contains an enzyme system which is responsible for oxidizing NADH2 and cytochrome B5. Now, your question can be, what does NADH2 oxidize to form? I am particularly mentioning about questions because I will be interested, and it will be my opportunity if you just put them in the comment box for me to help you better, and I'll certainly put my answers back there. Now, the inner mitochondrial membrane is highly folded, and these folds go up into the matrix. These folds are known as the cristae, also known as the mitochondrial crest. When a particular membrane is thrown up into so many folds, it is quite clear that the surface area of this membrane is increased. Now, when the surface area is increased and the mitochondrial chamber is thus divided into so many subchambers, the work done here is much more than had it been just a single membrane without any infoldings. The inner face, as we can see, I have just said that they are studded with elementary particles or oxysomes, and this membrane is selectively permeable. Alright, so now the inner membrane is highly folded, as we have said. Now we will come into the outer chamber, and which is the intermembranous or perichondrial space. The space between the outer membrane and the inner membrane is known as the perichondrial space. The perichondrial space is the space which is along the periphery of the mitochondrion, and this is known as the perichondrial space, and it is filled with proteins. It is filled with enzymes. Enzymes, as we know, they are proteins as well. The inner chamber is enclosed within the inner membrane. It is filled with gel-like homogeneous mitochondrial matrix. This matrix contains high concentration of hundreds of enzymes. It contains a high concentration of enzymes which work for the Krebs TCA cycle, Krebs tricarboxylic acid cycle. this Krebs cycle is a part of the respiratory process, the respiration process. The first part of the respiration process being carried out in the cytoplasm or the cytosol, which is known as glycolysis, and this part, the final part, it takes place in the inner chamber of the it takes place in the mitochondrial matrix, where the enzymes for the Krebs TCA cycle are there. Then there are some lipids here. There is a nucleoid, I would call it. It is similar to the nucleoid which is present in the prokaryotic cells. It is a blob of nucleic acid. It's a circular DNA molecule, and it also contains ribosomes. It contains tRNA molecules and enzymes for the functioning of the mitochondrial genes. Now, the elementary particles, as we see here, we have the elementary particles, the distribution, the mitochondrial fold, the elementary particles, as we can see. Now, this elementary particle is actually, it has got a head piece, it has got a stalk. The head piece or the rounded head piece is also known as the F1 subunit. The stalk is known as the F0 subunit. I'll repeat. When we are talking about the elementary particles or the oxysomes, we can see that there is a blob head, a rounded head, a little tiny rounded head, and a stalk. The head is also known as the F1 subunit, and the stalk is known as the F0 subunit. These two subunits, the F0 and the F1 subunits, together function as ATP synthetase or ATPase, thus leading to the formation of the ATP molecule, because this enzyme catalyzes the coupling of the oxidative phosphorylation process. So, I'll just repeat, the F0 and the F1 subunit combination, it functions as ATP synthetase. You can understand from the name of the enzyme that this helps in the synthesis of ATP, and the process of oxidative phosphorylation leads to the formation of ATP. Oxidative phosphorylation is a process which requires the raw materials ADP, that is adenosine diphosphate and inorganic phosphate. These join to form, that is, these get coupled up to form the ATP molecule, which is adenosine triphosphate. Okay, now I will discuss the mitochondria. They are autonomous or semi-autonomous organelles. What is the reason? They have got, that is, we know that autonomous bodies have their own set of rules governing their own functioning. Mitochondrion is similarly working on that principle. The reason is that it has got its own set of genes to guide its functioning. I've just talked about the molecule of DNA which is present in the mitochondrial matrix, and this DNA molecule is more or less similar to the prokaryotic nucleoid. I think I had said it then also when I began talking about it. Incidentally, these mitochondria, they are self-perpetuating. Why? Because it has got its own set of genes. It has got its it is self-perpetuating. Existing mitochondria divide and give rise to new mitochondria. Incidentally, the mitochondria are also regarded as the intracellular, that is something which occurs inside the cell, intracellular parasitic prokaryotes. And this is a sort of symbiotic, establishes a sort of symbiotic relationship with the cell. Now we know that the cell is housing the mitochondrion. The cell is giving shelter to the mitochondrion, it is allowing it to stay. What does the mitochondrion do in return for the cell? It helps in the oxidative phosphorylation, it helps in the completion of the Krebs cycle or the last stage of the respiratory cycle, the respiration. So it carries out, because it helps in respiration, it helps to complete the respiration chain, it is helping the cell and thus the living body. So it is regarded as a symbiotic relation. Now the mitochondria, they have they have got certain similarities with the bacteria. The bacteria, we know they are prokaryotic organisms. Let us just discuss a few points where we can see that the mitochondria is actually behaving like the bacteria. Now we have seen the inner membrane of the mitochondrion is thrown up into infoldings or mitochondrial crests. They are known as cristae also. Similarly, in the bacteria, they are thrown up into infoldings which are also known as mesosomes, and these house the respiratory chain enzymes. So definitely it is a perfect similarity which has been drawn here. Then we know the presence of the DNA. In both the cases, in the mitochondrion as well as the bacteria, in the mitochondria as well as the bacteria, there is a single DNA molecule which is there. How are they similar? It is the one present in the nucleic acid present, which is present in the mitochondria. They are without any boundary. They are not bounded by any membrane, in just the similar way as any prokaryotic cell such as the bacteria. We know that the bacteria houses nucleic acid which is not bounded by any membrane, and thus they are known as nucleoids. Well, they are similar. So here this is certainly a similarity. Then there are ribosomes which are present in the mitochondrion, and it is similar to the bacterial ribosomes. Now mitochondria, they have got mitochondria as well as the bacteria, they can replicate on their own. Thus we can call them autonomous, don't we? We are calling them autonomous. Why? Because they can replicate on their own. Now this replicating of one mitochondrion into the other is independent of the chromosome replication in the nucleus of the cell of which it is a part. I'll make it a bit clear. This mitochondrion is present inside a cell. This cell has got a nucleus which is the brain of the cell, and the cell works according to whatever the nucleus has to say, because the nucleus has got its own set of genes which are housed in the chromosomes. Now the mitochondria replicate and become new mitochondria. This is independent of the decision which is taken by the nucleus of the cell of which it is a part. All right. I'll now talk about the functions of the mitochondria. A very important function of mitochondria is that the mitochondrial membrane, the mitochondrial matrix carries on the Krebs cycle or the aerobic respiration. During this process, we know that the end products of respiration are carbon dioxide and water. How are they formed? That is, the food material is interacted with oxygen and carbon, and the ultimate end products are carbon dioxide and water. This is actually what takes place here. The glucose, the amino acids or the fatty acids, they are degraded into carbon dioxide and water. And with this happens when the latter, they react with oxygen. The energy liberated by these foodstuffs, we know, is it is just locked inside some energy rich molecules known as ATP or adenosine triphosphate, and it is utilized whenever it is necessary. Now, the second quite an important function of mitochondria is it helps in the formation of yolk. Where is yolk present? In the ovum. It helps in the formation of yolk in the developing ovum and are converted into some yolk storing bodies where the yolk are actually stored. So mitochondria helps in the formation of yolk. Mitochondrion also helps in the formation of elongated chains of fatty acids. And these are formed when the fatty acids are, attached to acetyl coenzyme A. Fatty acids, when they are attached to acetyl coenzyme A, they are elongated, and this happens in the mitochondria because these contain enzymes for this particular type of process to take place. Now, this chapter on mitochondria is actually has got, they have some certain important questions. I feel they've got innumerable questions though, but here I've just selected a few. What is the purpose of the enzyme system present in the outer mitochondrial membrane? Question 1. What is the purpose of the enzyme system present in the outer mitochondrial membrane? Question 2. Name some important constituents of the mitochondrial matrix. Question 3. What does the F0 and F1 subunit combination function as? I have discussed about the answers of all these questions. Please do recollect. And in case you have any problems, please put your problems in the comment box, which I'll definitely care to answer. Please do like, share and subscribe my channel. Thank you so much.